I'm here to introduce you why we need to have meiosis. So first off, we need to understand meiosis is different from mitosis, which result into identical cells. Mitosis is actually a type of cell division that result in 4 daughter cells, each have half of the numbers of the chromosomes of the parent cell. So why it's important? Because we want our offsprings to be different from ourselves, so that they can have a lower chance from suffering of the genetic diseases. Say if we have identical offspring as ourselves, then there is a very high chance that they will suffer from certain disease. With different chromosomes and different types of cells, they're able to avoid those types of diseases.
Hello everyone. So now that we have explained what meiosis is and what is its purpose, now we're gonna go through meiosis phase 1 and meiosis phase 2. And what I'm going to explain is phase 1. So during phase 1, what happens first is the interphase. During the interphase, the cell duplicates, the centrosomes and the DNA is all loose. It is not tightly wound up into the chromosomes. However, later on during the interphase, as it goes through the S phase, its DNA is duplicated and it is formed as sister chromatids, and now we have the first chromosome and the second chromosome, and which basically this is the mom's chromosome and the dad's chromosome. And then we move into prophase 1. During prophase 1, what happens is synapsis, where chromosome 1 from mom and 1 from dad are coming together and they're switching parts of the chromatids, which this is called crossing over. So basically what happens is that they are giving segments from each one to each other. For example, the mom, that in this part it's the blue chromosome, will give a segment to the dad's chromosome, which is the brown one, and vice versa. Same thing will happen here and here, and same thing will happen for the chromosome 2. Now that we have switched segments from each of the mom's and the dad's chromosomes, we have different chromosomes. Now we move on into metaphase 1, which is when both chromosomes are aligned up in the middle of the cell at the metaphase plate, and they have an independent orientation, which means that they have 4 possible probabilities where they can be placed. For example, here we have 2 chromosomes and 2 sister chromatids, which makes 4 probabilities or possibilities. Which means that this one can go this way, or this one this way, and this one this way, this one that way. And that's what it means. That's what it is, the independent orientation. And for example, in the human body, it would be 2 raised to the 32, I believe, because it has 32 chromosomes or
23. Like 23 pair.
or 23, something like that. It's just an example. Then we move on into wait, never mind. Again, in the metaphase, the spindles are going to attach to the centromere of each of the homologous chromosomes. As you can see, they're attaching to each homologous chromosomes. And then during anaphase, the homologous chromosomes are being pulled apart by the spindles right to the sides of the cells, which means that they're going to either cell. And then we move on to telophase 1, which means that there is reforming a new nuclide at each side. As we can see, there's a nuclide at each side, and we've now divided the nuclide, which is meiosis. As you can see, now it's divided, it's meiosis, but finally we had to cut it up into two cells, which is so then the cells has to be divided into two, and that is called cytokinesis. And basically this is meiosis 1.
Hello everyone, I'll be explaining meiosis 2. So meiosis 2 is very similar to meiosis 1 and also mitosis with both phases. So we start out with two diploid cells with the DNA that is in the nuclear membrane. Then we move to prophase where the nuclear membrane dissolves. Then as we move to metaphase, we see the chromosomes lining up and the spindle fibers attaching. And then as we move into anaphase, the chromosomes are pulled apart and getting ready for cytokinesis. And then as we move down into telophase, the nuclear membrane reforms, and now we have 4 haploid gametes after cytokinesis. So the haploid gametes have a half set of DNA. So half, one of these will come from the mother, one will come from the father, and they both have half. So that makes a full set of DNA.
No. What is it for?
Alright, so half will come from the mother's egg, and then half will come from the father's sperm.